Welcome to Biology at Priyojit, my dear viewers. Today we'll explore cloning vectors, the powerful DNA carriers that make genetic engineering possible, from plasmids and bacteriophages to advanced systems like BAC, YAC, MAC, and expression vectors. We will must understand their key characteristics, their carrying capacity and applications. So get ready to see how these vectors help scientists clone, maintain and express genes with precision in modern biotechnology. To introduce the gene cloning, we must say that gene cloning is a major breakthrough in biotechnology. The success of gene cloning mainly depends on the proper selection of a cloning vector. Without a suitable vector, cloning becomes ineffective. What is gene cloning? Gene cloning is fundamentally is the process of isolating a specific gene from a donor organism, inserting it into a vector, replicating it inside a suitable host cell. What are the applications of gene cloning? The image here showing the basic process of gene cloning. So the applications include DNA sequencing and genetic research, gene therapy for treating various diseases, development of improved crop varieties, production of antibiotics, vaccines and vitamins, industrial and pharmaceutical biotechnology. Steps involved in gene cloning: a fragment of DNA containing the gene to be cloned is inserted into a circular DNA molecule called a vector. So vector is a carrier molecule that only can carry your gene of interest or your DNA of interest to produce a recombinant DNA molecule. So the vector transports the gene into a host cell, which is usually a bacterium, although other types of living cell can be used. Within the host cell, the vector multiplies, producing numerous identical copies not only of itself but also of the gene that it carries. When the host cell divides, copies of the recombinant DNA molecule are passed to the progeny and further vector replication takes place. After a large number of cell divisions, a colony or clone of identical host cells is produced. Each cell in the clone contains one or more copies of the recombinant DNA molecule. The gene carried by the recombinant molecule is now said to be cloned. Now we should start regarding the cloning vectors. We must know what are the cloning vectors, their definitions and characteristics. Cloning vectors are basically DNA molecules used to carry foreign DNA fragments into host cells for replication and or expression. An ideal cloning vector has a selectable marker, that is antibiotic resistance marker, multiple cloning site or MCS with unique restriction sites, origin of replication, that is oricite, and ability to accept foreign DNA without lethality. So the first cloning vector is the plasmid. So we have to know what are plasmids. Plasmids are basically small circular double-stranded DNA molecules. They exist independently of the bacterial chromosome. They carry useful genes for the host bacterium. Importance of plasmid. So these are the image showing the required features of plasmid. Importance of plasmid: it often carry antibiotic resistance genes, ampicillin and or chloramphenicol. In laboratory, antibiotic resistance is used as a selectable marker to identify bacteria carrying plasmids. Replication of plasmids have their own replication origin. They can replicate independently inside the bacterial cell. Small plasmids used host enzymes for replication. Larger plasmids may carry their own replication genes. Some plasmids can integrate into the bacterial chromosome called episome. So this is the plasmid size and copy number. The size ranges of plasmid from 1 kb to 250 kb. Cloning vectors are usually less than 10 kb. Copy number of plasmid molecules per cell, that is stringent plasmids have low copy number, that is 1 to 2 per cell, and relaxed plasmids have high copy number, that is 50 or more per cell. High copy number plasmids are best for cloning. So we must know what are the classification of plasmid or types of plasmid based on function. The first of all is the fertility plasmid that carry TRA genes and help in conjugation process. Example F plasmid of E. coli. R or plasmid or resistance plasmid that provide resistance to antibiotics or heavy metals. They are important in clinical microbiology. For example RP4. Col plasmids, they produce collicins that kill other bacteria like col E1. Degradative plasmids help bacteria degrade unusual substances. Example tall plasmid. Virulence plasmid, they confer pathogenicity, that is plasmid of agrobacterium. Plasmids in other organisms, they are very common in bacteria but rare in eukaryotes. Best known eukaryotic plasmid is 2 micron plasmid of yeast. They are useful in making yeast colony vectors. Their most higher organisms do not contain plasmid. So plasmid, we know the beginning of among the cloning vectors, and it was discovered early 70s. In 1973, Cohen and Boyer first successful recombinant DNA using plasmid. Uh, how they are discovered? Plasmids are naturally found in bacteria as extra chromosomal circular DNA. Scientists noticed they could replicate independently. They carry genes like antibiotic resistance. Why they became the first vectors? Because plasmids are easy to isolate. They have small size. They have a high copy number. So limitation discovered later. They could carry only small DNA fragments, that is maximum 10 kb. Now we should go for bacteriophage lambda, need for bigger inserts. It was developed in mid 1970s. Why plasmids were not enough? Genomic studies needed larger DNA fragments, so plasmids had size limitation. Discovery insight: lambdaphage naturally infects E. coli. It has cosmids which is efficient for DNA packaging. Innovation: no, they are not essential lambda genes removed. Foreign DNA inserted without affecting packaging. As a result, cloning of 99 to 23 kb fragments, they have high infection efficiency. Limitation: they are still not large enough for the whole genome. Bacteriophage lambda as a cloning vector has some general features. Lambdaphage infects E. coli and is widely used as a cloning vector. It has both lytic and lysogenic life cycles. DNA inside phage particle is linear, but it ends up 12 BP cohesive or cos sequences. Inside E. coli, the cos ends anneal and ligate, forming a circular DNA. DNA packaging capacity: only DNA of 37 to 52 kb can be packaged into lambda phage head. Small inserts can be added directly. For large inserts, part of lambda genome must be removed. Lambda genome organization: the left region of the genome are essential for head and tail structural proteins. Right region, they have genes for replication and lysis. Middle regions is approximately 15 kb length. They are non-essential for lytic growth, but they contain genes for integration and recombination, and they can be replaced with 23 kb foreign DNA. Lambda replacement vectors: foreign DNA replaces the middle region. Such vectors cannot integrate into host chromosome on their own, so helper phage is required for integration and lysogen formation. In vitro packaging of the lambda phage DNA: recombinant lambda DNA with foreign insert is linear with cos end, and efficient entry into E. coli requires in vitro packaging. Cosmid, that is a hybrid solution, and it was developed in late 1970s. Problem: plasmids are small inserts, lambda phage are medium inserts. Scientific idea: we can combine them, the plasmid replication ability, lambda coside for packaging. Discovery: creation of cosmids. Cloning capacity increased to 30 to 45 kb. Uses early genomic libraries. Limitation: there is no expression of genes, only cloning purposes. Cosmid vectors. What are cosmids? Cosmids are basically hybrid vectors, plasmid carrying lambda phage cos site. It exists as multi-copy plasmids in E. coli. This is the image showing the cosmid. It do not carry lambda genes, only the cos site. Why cosmid vectors are used? Lambda packaging does not require lambda genes, only cosmids. It allows cloning of very large DNA fragments and combine the advantages of both the plasmid and lambda phage. Key features: it has cosmids from the lambda phage enable packaging into the phage head. It has oricite for replication. It has a selectable marker. It has a cloning with cosmid. Cosmid is linearized, producing cos ends. Foreign DNA and cosmid are cut using same restriction enzyme or different enzymes producing compatible sticky ends. That is example for BAM-H1 or MBO1, etc. Target DNA is often partially digested. It helps isolate large genomic fragments and ensure intact gene is present even if a site lies within the gene. Packaging and infection. Ligation produces DNA with cos sites at both ends. Recombinant cosmid DNA is packaged in vitro into lambda heads. Packaged particles infect E. coli efficiently. Where inside the cell, the cosmid behaves like a plasmid. Insert size small cosmid, that is approximately 4 kb, can carry up to 45 kb foreign DNA. Key advantage: it is ideal for genomic libraries and cloning large DNA fragments. M13 and Phagemids, there is the rise of DNA sequencing 1980s. Why needed? Sanger sequencing required single-stranded DNA. So filamentous Phage M13 produces ssDNA. Later plasmids with M13 origin produced phagemid. Applications: DNA sequencing, site-directed mutagenesis, etc. But it was a limitation, that is small insert size like plasmid. So what are the M13 vectors? M13 vectors are derived from the filamentous bacteriophage M13, which infects E. coli strains carrying the F plus or F plus cells. Key features: single-stranded DNA or ssDNA genome. M13 exists mainly as circular ssDNA which enters E. coli. Inside host cell, it is converted to double-stranded replicative form or RF, which replicates like plasmid. Later, the ssDNA is produced and packaged into phage particles. Filamentous phage, it is long, thin, non-lytic phage. Phage particles are released without killing the host cell. Origin of replication: special F1 origin enables ssDNA synthesis and their packaging. Insertional cloning site. Foreign DNA is inserted into the intergenic region. It does not disrupt essential phage genes. Small insert capacity, typically approximately 5 to 7 kilobase per. There is no cos site, and the packaging occurs by extrusion through the bacterial membrane, not by head tail assembly. Selectable markers: the modified M13 vectors carry LAGZ, a fragment for blue-white screening. It requires F plus host. Infection occurs via F plus. Uses of M13 vectors: DNA sequencing by means of Sanger method, site-directed mutagenesis, phage display technology, and preparation of ssDNA probes. What are the advantages of M13 vectors? It provides high quality ssDNA, non-lytic to the host cell, and it is ideal for sequencing work. What are the limitations of M13 vectors? It has low insert capacity. It is not suitable for large DNA cloning. It requires A plus E. coli strains. So this is the genome of M13 phage. Plasmid. Why were plasmids invented over M13 bacteriophage vectors? Plasmids were invented to overcome the practical limitations of M13 bacteriophage vectors while retaining their most useful feature, that is single-stranded DNA production. So this is the image showing the general structure of plasmid. Limitations of M13 vectors are exist only as phage, low DNA yield, small insert size, requires continuous infection, and it has limited selectable markers. So what is the concept behind plasmid development? Scientists ask, can we combine the easy of plasmids with the ssDNA advantage of M13? So the plasmids were the answer. Advantages of plasmid over M13 vectors. This is the table showing their advantage. This is plasmid. Plasmid is basically a hybrid vectors that combine the properties of plasmid and the filamentous bacteriophage M13. Its size is 5 to 15 kb insert. Plasmid key features: It is circular double-stranded DNA like plasmids. It contains plasmid origin of replication or ORI. Replication is performed as plasmid F1 or M13 phage origin. Production of single-stranded DNA. It has selectable marker genes like antibiotic resistance, possesses multiple cloning site or MCS. It requires a helper phage to produce ssDNA. It can exist in two forms: DS, that is double-stranded DNA or plasmid form, and ssDNA or single-stranded DNA, that is phage form. Insert size is 5 to 15 kilobase per. Uses DNA sequencing, site-directed mutagenesis, phage display, hybridization, probes formation, etc. So this is the general structure of plasmid. BAC stability became important in late 1980s. New challenge came. Cosmids and Lambda vectors were unstable for large DNA. Scientific breakthrough, use of F factor plasmid of E. coli, discovery, that is bacterial artificial chromosome. Its advantages showed very stable structure, low copy number, and its insert size was 100 to 300 kilobase per, and it is mainly used in human genome project. So this is the structure of a bacterial artificial chromosome. It is derived from F factor of E. coli. 100 to 300 kilobase per. Key features: it has single copy, so it has stable propagation, ORI form F factor, and it has a chloramphenicol resistance. It is used in genome mapping, large genomic libraries formation. YAC, it is a cloning extremely large DNA. It was developed in 1980s. Why BAC was still not enough? Some genes and regulatory regions are megabase sized. Idea: mimic a real chromosome. So Yeast Artificial Chromosome or YACs were developed, and it has insert size up to 1 megabase pair or more, but the limitation: high recombination and less stable than BAC. So this is a structure of YAC vector. It is vector based on Yeast chromosomal elements. Its insert size is 100 to more than 1000 kilobase pair. Key features: it has ORI site, it has centromere, it has telomere at ends, it has selectable markers. It is used for cloning very large DNA pieces, and it functions like a real chromosome in yeast. MAC, that is expression in mammals, 1990s to 2000. Bacterial or yeast systems cannot properly express mammalian genes. So solution: create a artificial chromosomes of for mammalian cells. So mammalian artificial chromosomes were discovered, and its applications were gene therapy and functional genomics. So artificial chromosomes functioning in mammalian cell is the mammalian artificial chromosome. This is the image showing the mammalian or human artificial chromosome. It is megabase range. It is the biggest insert. Key features: it has centromere, telomere, replicon origins, and it is stable. It has low disruption of host genome. It is basically used in gene therapy and functional genomics. Expression vectors. Why expression vector is from cloning to protein production? Why expression vectors were needed? That is why. What is the evolutionary need? Cloning vectors only carry and replicate foreign DNA, so inserted genes often remain silent or produce very little protein. Scientists realized that cloning alone is not enough, genes must be expressed. So there was a growing need for large scale production of functional proteins, high demand for insulin, growth hormones, vaccines, and industrial enzymes. So hence expression vectors evolved to ensure efficient gene expression and protein expression. This is the image showing the expression vector, and this is the process how cloning vector is converted into expression vector. This is the table showing the evolution of different vectors and why they were discovered one by one. So expression vectors is basically vectors designed not just to carry DNA but to express proteins. Expression vectors were developed by modifying cloning vectors to include gene expression control element. It has following characteristics like origin of replication for to replicate inside the host cells. It has a strong promoter which helps to start transcription, ensure high level of gene expression. It has a ribosome binding site which is required for protein synthesis. It has multiple cloning site that contains many restriction enzyme sites which makes the insertion foreign gene easy and accurate. It has selectable marker genes which helps identify transformed cells. It has transcription terminator site which signals end of transcription. Fusion tags, it has a short sequences attached to the protein, helping easy purification and detection of the expressed protein. It has host compatibility, that is vector is designed according to the host like bacterial, yeast, plant or mammalian cells. So in conclusion, we can say that cloning vectors are indispensable tool of genetic engineering that enable the stable insertion, maintenance and expression of foreign DNA. From simple plasmids and bacteriophages to advanced systems like BAC, YAC, MAC and expression vectors, each vector is designed with unique characteristics to suit specific cloning and expression needs. Together they form the backbone of modern biotechnology, driving advance in research, medicine and industry. Keep exploring the fascinating tools of modern biology or biotechnology. Thank you.